Welcome to a new module in our immunology course where we are going to focus on antigen presentation and the biology and genetics of MHC. And in this first couple of videos, we're going to talk about more cell biology in terms of how is it that we process antigens and actually get them onto the MHC molecule so that they can be presented to T cells. And in this very first video, we'll discuss the processing of what we call endogenous antigens, which usually are presented by MHC class 1 to cytotoxic T cells. So this table from the Janeway textbook is a pretty good resource, I would say, into thinking about the different types of antigens and how they fit into an overall immune response. So as you can see here, pathogens can be found in different locations, either inside or sort of relative to the cell. And they need to be processed, or antigens or peptides associated with pathogens need to be processed in different ways in order to actually be loaded onto MHC molecules to be presented. So if we just kind of go across here, you can see that as a class, we think of pathogens that tend to exist in the cytosol as a sort of set of pathogens that usually end up being presented via MHC class 1. Of course, that means that they are being presented to CD8 T cells, the effector mechanism of which tends to be cell death. So cells that are actively infected with some kind of intracellular pathogen, this is very commonly a virus or some kind of intracellular bacteria, they tend to be targeted for death because they present antigens on MHC class 1 to CD8 T cells. In contrast, there are pathogens that we might think of as intravesicular pathogens. Maybe they are pathogens that actually tend to live inside the cell, but they live inside vesicles rather than the cytoplasm. Or perhaps they are brought into the cell through vesicular trafficking processes like endocytosis or phagocytosis. In either case, we think of those pathogens as more extrinsic to the cell. Even though they sort of live inside the cell in some cases, they're not in the cytoplasm directly. They're still sort of sequestered off from the cytoplasm inside those vesicular compartments. And again, this involves different types of pathogens, other types of bacteria, as well as funguses and some small parasites. And instead of being presented, or instead of their antigens being presented on MHC class 1, these types of antigens tend to be presented on MHC class 2, which means that they are presented to helper CD4 T cells. And CD4 T cells, as you may remember, instead of killing their targets, instead, mostly their effector mechanism mostly is the production of cytokines, which sort of activate and unlock other effector mechanisms in other cell types in the environment. And so one example of that would be within macrophages. When macrophages present antigens to CD4 T cells, the T cells in turn produce cytokines that activate those macrophages in ways that increases their ability to kill intravesicular pathogens like bacteria and parasites. So there's one additional sort of set of antigens that we can think of, and those are not really derived, or not really whole organisms or proteins derived directly from organisms. Um, but frequently these are just sort of soluble antigens or soluble pieces of pathogens that can especially be recognized by B cells and that induce BCR cross-linking, as you can see here. And BCR cross-linking actually induces the process of endocytosis. And so B cells, remember, are professional antigen-presenting cells, and that means that they typically, or they're capable anyway, of presenting antigen on MHC class 2 as well. And a point that's that we haven't really covered yet, but B cell activation actually requires the participation of CD4 T cells. And so in order for T cells to be involved in B cell activation, there's some sort of crosstalk that happens. The B cell senses its antigen, it internalizes that antigen, it degrades it, and then it presents that antigen to a CD4 T cell. And in return, that CD4 T cell secretes cytokines, which signal back onto the B cell in ways that sort of activates its ability to produce immunoglobulins, to undergo class switching, and so on. And in so doing, sort of unlocks all the effector mechanisms associated with antibody secretion. So just a few categories here of different types of pathogens, the types of T cells they tend to activate, and the roles for each in various types of immune responses. So we're going to sort of go through these each in this module, but we're going to focus in this first video on the processing of cytosolic or endogenous pathogens, which again are going to be presented to CD8 T cells. But as we think about differences in the way that we present antigen via MHC class 1 and MHC class 2, let's remind ourselves of the structure of both of these molecules. Um, you may recall, as we said in a previous video, that they are similar molecules, but they have some structural differences. So for example, MHC class 1, its peptide binding groove is formed by its alpha 1 and alpha 2 domains. And you can see here this sort of top-down structural diagram. There are helices associated with both of those domains, and the peptide antigen sort of fits right in between those helices. That's what's shown here in orange. And as you can see in the table, the peptide binding groove of MHC class 1 is closed at both ends. It sort of it actually forms a nice little basket almost that is sort of closed all the way around. And the consequence of that is that peptides can fit inside it, but they don't tend to sort of have the leeway to sort of hang over the sides. The closure of the grooves sort of restricts the potential size that can fit within the peptide binding groove of MHC class 1. And in fact, that size is about 8 to 10 amino acids, which is actually quite short. The antigens presented by MHC are very small peptide sequences. MHC class 2 is not too different, but as you can see, its peptide binding groove is made up of the alpha 1 domain of its alpha chain, as well as the beta 1 domain of its beta chain. And its structure is slightly different. It, instead of being closed at both ends, is open at both ends. And the reason that matters is that that allows it to accommodate a slightly larger range of amino acids, as well as an overall different size of amino acid chains, so somewhere between 13 and 18 amino acids. Tend to be the size of peptides produced on presented on MHC class 2. So you can see already that the peptides presented by both are slightly different, and that's going to sort of convey to us the idea that they are probably processed in different ways, because 8 to 10 and 13 to 18 amino acid chains don't really exist in nature all that much. Most proteins are much larger, suggesting that proteins associated with pathogens have to be degraded or processed in some way to get down to these small amino acid fragments or fragments of polypeptide chains. And so we'll be sort of seeing how is it that we generate these two sort of unique sets of amino acids that are associated with presentation on both classes of MHC. So focusing on endogenous antigen presentation via MHC class 1, if we want to take a sort of overall view of what that looks like, we're going to start with a cytosolic protein. Again, probably that's going to be derived from a pathogen that lives in the cytoplasm, although this process also occurs for self-proteins. And we'll sort of unpack what happens to self-peptides a little bit later as we go through the course. But for the purposes of today's lecture, let's just imagine that this is a protein associated with an intracellular bacterium or a virus. Like most proteins in the cell period, those proteins will sort of have a certain lifespan and will ultimately be degraded. They'll be targeted for ubiquitination. The ubiquitination of these proteins targets them for processing by a structure called the immunoproteasome. Now, you may have heard of a proteasome before. Most cells have what is called a constitutive proteasome, which is responsible for degrading and recycling proteins that have been ubiquitinated. The immunoproteasome is closely related, but it is made up of some special subunits, which process cytosolic proteins in a way that is especially good for generating the lengths of peptides that we need to be presented on MHC class 1. So there's a kind of specialized proteasome called the immunoproteasome that is going to degrade those ubiquitinated cytosolic proteins into small peptide fragments. Those peptide fragments are then going to be shuttled out of the cytoplasm into the rough endoplasmic reticulum, the lumen of the rough endoplasmic reticulum, through a transporter molecule called TAP. Once they are inside the endoplasmic reticulum, there they can be loaded into the peptide binding groove of MHC class 1. And following successful loading of antigen peptides, MHC class 1 is then sort of released from the ER and traffics through the Golgi and the sort of general vesicular system, and vesicles containing MHC class 1 peptide complexes end up fusing with the plasma membrane in such a way that they are then displayed on the extracellular face of the plasma membrane. And that is there that they can interact with T cell receptors. And of course, that interaction, as we've seen, also requires binding to the co-receptor CD8. So this is kind of the whole life cycle of an endogenous antigen, and we're going to, in subsequent slides, sort of go through each of these steps in a little bit more detail. But this is this sort of overall picture of what we want to discuss. So let's start with, or start at the beginning. I said that endogenous antigens are derived from cytosolic proteins that have been ubiquitinated, and you can see an example of that here, some kind of protein with a bunch of ubiquitin sticking off the end. And they are going to be degraded by the immunoproteasome, which sort of has two distinct sort of components. A 19S regulatory particle, or RP, and these sort of form the caps of the immunoproteasome, as you can see. And these cap structures, these 19S structures, are important for recognizing ubiquitinated proteins and sort of, pulling them in, binding to them, and then also kind of threading the polypeptide chain into the core of this complex. And the core of this complex is made up of a 20S subunit, which actually has enzymatic activity, has protease activity, it actually has a variety of protease activities. Some of its subunits cleave acidic amino acid residues, some cleave basic, and so on and so forth. So there's multiple ways that it can chop the protein up. But when it does so, it leaves a variety of peptide fragments, which themselves have a variety of lengths. Some of them are already the correct length that we need, but many of them are not. So some of these peptides will require further processing. This initial stage of fragmentation via the immunoproteasome is a little bit of a rough job. We just sort of want to chop our big protein up into much smaller pieces so that we can work with it. But this is the nutshell version of how the immunoproteasome accomplishes that. But once these proteins have been degraded by the immunoproteasome, we have these peptides sort of floating around in the cytoplasm. But of course, we need to get them into the rough ER, which is where MHC class 1 is. And the reason that the MHC class 1 is in the rough ER is because it is there being folded. It is associated with chaperone molecules because it has just recently been translated. And like basically all proteins, they need to be translated by ribosomes, and they need the help of chaperone molecules to fold correctly within the ER. And MHC class 1 is no exception to that. And a key point is that it's actually peptide, antigenic peptide, that is required for the full folding and full stability of MHC class 1. So we need to bring the peptide into association with MHC class 1 where it is being folded in the rough ER in order for that full process to sort of play out. So the peptides, in order to get where they need to go, need to be shuttled out of the cytoplasm into the lumen of the ER, and that is accomplished through a transporter enzyme called transporter associated with antigen processing, or TAP. And TAP, through an energy-dependent, an ATP-dependent process, sort of pumps these peptides out of the cytoplasm into the lumen of the rough ER. And these peptides encounter MHC class 1 as it is being folded with the help of molecules that make up something we call the peptide loading complex, which is a set of chaperone molecules that hold the MHC class 1 molecule together until the endogenous antigen can be loaded. And once it is loaded into the peptide binding groove, again, the process is complete. It can be sort of released, liberated from the ER, and sort of encapsulated in a vesicle, which can be targeted to the cell surface so that it can be displayed and accomplish the process of antigen presentation. Let's zoom in now on another one of these pieces, though, and that is the peptide loading complex, that set of chaperone molecules which facilitate the folding of MHC class 1, and as well as the process of antigen loading. So the PLC has a few major members. I've given you some of the most important here on this slide. And we'll sort of start at the beginning. Recall that MHC class 1 is made up primarily of an alpha chain, MHC1 alpha, as well as a sort of accessory subunit called beta-2 microglobulin, which is synthesized separately, and the two things have to be brought together to form the functional MHC isoform. the MHC class 1 alpha chain is highly unstable on its own without beta 2 microglobulin. And so the first chaperone that's important is a molecule called calnexin, and it stabilizes the MHC class 1 alpha chain until it associates with beta 2 microglobulin, which gives the molecule a little bit more stability, but not nearly enough. It still is not completely stable until it has a peptide in its binding groove. So the successful sort of conjugation of MHC class 1 with beta 2 microglobulin allows calnexin to sort of pass the molecules off to some additional chaperone molecules, which are listed here. We have calreticulin, which it serves as an important chaperone for holding the alpha chain and beta 2 microglobulin together, as well as ERP57, which facilitates some of the processes of folding, as well as serves as a physical link with a very important chaperone called tapasin. And tapasin's main function, you can see that it has tap in its name, is to really serve as a physical bridge, a physical kind of link to tap. And so because tapasin will bind or associate directly with tap, it brings this whole complex into direct physical apposition to tap. And that's important because these peptides are coming in through the tap transporter. And we want the MHC class 1 molecule to be right nearby so that it can sort of catch the peptides as they're coming in. And if those peptides successfully sort of get into the right location, they're able to bind to the peptide binding groove. This structure is stabilized, and that allows the chaperone molecules to dissociate because now MHC class 1 has been fully successfully folded. And their job is complete. And if we look at a sort of structural diagram of the chaperone molecules, I really love this image just because I think it just sort of underscores exactly what is happening. You can see that the combination of tapasin, ERP57, and calreticulin form this nice little pocket which sort of hugs the MHC molecule as it's being folded and prevents it from falling apart prior to the loading of peptide into its binding groove. And so this is a very effective mechanism of chaperoning that they accomplish there in the rough ER. Okay, so one additional point that I want to make about the things that need to happen in order for antigen to be loaded into the peptide binding groove. I said before that the peptides that come out of the immunoproteasome are a variety of lengths because the immunoproteasome is using a variety of different protease subunits, all of which are sort of acting on their own, not necessarily in a highly concerted way. There's just a lot of sort of chopping up going on. And the immunoproteasome tends to generate peptides anywhere between 3 and 22 amino acids in length. And remember that we need something more like in the realm of 8 to 10. And while 8 to 12 amino acid lengths are the most common output of the immunoproteasome, the fact that we can get much shorter and much longer amino acid lengths is sort of suboptimal. That means that many of the peptides that the immunoproteasome puts out are actually unusable as antigens without further processing. Now, generally, those that are too short are not really used any further. You can't really add amino acids to a peptide that you've cut to be too short. But the ones that are too long can undergo some additional selection and a process called editing to get them down to the 8 to 10 range that we need. So a couple of selection processes happen. One is that TAP itself is actually selective only for peptides that are in the 8 to 16 amino acid length. So what this means is that TAP actually excludes peptides that are too short and leaves them in the cytoplasm where they can eventually be degraded and recycled. And it also excludes those peptides which are way too long, between 16 and 22 amino acids. So that means that the peptides that do make it into the rough ER are already enriched to be between 8 and 16 amino acids in length, which is a much better range than what we started with. However, remember that we still want a smaller range than that, 8 to 10. And so there's an additional enzyme in the rough endoplasmic reticulum called ERAP, ER aminopeptidase, associated with antigen processing. And you can tell that the aminopeptidase, that it's probably a protease of some kind. And it does something that we call editing of the peptides, where it sort of cleaves them at their N-terminal ends in ways that sort of trims them down to 8 to 10 amino acids, those that were longer than that to start with. And the key point here is that this further enriches the sets of peptides that are in the rough ER to be the correct length so that they have a high affinity for MHC class 1. It's not that, an amino acid or a peptide with 11 amino acids would never bind to MHC class 1, but it would do so with much less affinity because it's just a little bit too big to fit snugly in the antigen binding groove, if you will. And so the process of editing allows us to get into that sweet spot of length so that peptides associate with really high affinity with MHC molecules. That's important because we've said that the peptide stabilizes the MHC molecule. And so if you had a low affinity peptide, it might initially bind, and maybe it would bind long enough for the PLC complex to let go. But if the affinity is low, it actually might dissociate. There would be a dissociation constant associated with that interaction, which actually may um, not be all that strong. And so if a peptide binds with low affinity and then later dissociates, that MHC class 1 molecule is going to fall apart. It's going to unfold. And that would not be very helpful. That would be sort of a waste of an MHC molecule. Similarly, it may make it all the way to the cell surface, but if the peptide doesn't sort of hang on long enough, then it may dissociate prior to successful presentation to the TCR. So the 8 to 10 amino acids, as I said, are the sweet spot where we have the highest affinity for MHC class 1, and that is going to yield, what is essentially a permanent holding on, or at least a very long-lived interaction between those peptides and MHC class 1, which is going to ensure the stability of that interaction and the longevity of that interaction to make sure that those peptides actually make it to the T cell, if one that recognizes that antigen happens to be in the vicinity. Okay, so let's summarize this initial discussion of endogenous antigen presentation or processing. So we saw that there are distinct pathways that antigens can be processed through, and that generally intracellular, or what we might call endogenous antigens, are presented via MHC class 1, whereas extracellular or exogenous antigens are presented by MHC class 2, which will be the subject of our next video. Endogenous antigens, however, are processed primarily through proteolytic degradation by the immunoproteasome complex, which will chew up ubiquitinated proteins in the cytoplasm. And once they are cleaved by the immunoproteasome, they are transported into the lumen of the rough ER by the transporter molecule TAP. We saw that MHC class 1 requires several chaperones in a complex called the PLC in order to fold correctly and also to successfully load antigen. Some examples of those were molecules like calnexin, which stabilized the MHC class 1 alpha chain until the addition of beta 2 microglobulin, as well as other members like calreticulin and tapasin, which were important for folding as well as bringing the molecule into close association with TAP so that it could sort of catch antigens as they come into the ER. But assuming all that happens, antigen loading occurs, and it is further facilitated by peptide editing accomplished by the enzyme ERAP, which trims peptides to the most appropriate, the most high affinity length of 8 to 10 amino acids. And once all of these steps are complete, the complex of MHC class 1 and antigen traffics to the cell surface where it can successfully present that antigen to CD8 T cells. So that covers it for endogenous antigen presentation. In our next video, we'll switch our attention to exogenous antigen processing. See you in that video.